Cool. So yeah, I guess as you all saw in Slack, I'll be talking about learning rules in the brain today. And this is, I think, what's going to be kind of part one of probably like a two-part presentation. And unfortunately, Rami, the second part I'll probably first give in kind of September or something after our other researchers, Jose and Scott, join. But yeah, I'm sure there'll be a chance where you can see it as well. Yeah, the general kind of aim is to cover the diversity of learning rules in the brain. But what I'm really going to focus on is kind of the computational details and what the kind of implications of these are. And of course, the main motivation for this is to kind of explore the significance of these for Monty and hopefully kind of trigger some inspiration or kind of ideas about how we might use these either now or in the future. And yeah, obviously stop me if anyone has any questions. Yeah, I guess what the presentation is not is kind of, yeah, I'm not going to cover the full diversity of learning in the brain. And in particular, I'm not going to be covering it at a particularly biological level. Fortunately, I kind of discovered this presentation from Jeremy Forrest before I had put together too many slides. And I thought that was a really nice summary of all the kind of different learning rules from a kind of biological perspective. And so I would definitely encourage everyone to watch that if you've not had a chance. And yeah, I mean, you could watch it after this as well.
I'm a little confused. If it's about learning in the brain, but you're not going to focus on the biology, is it really?
I think it'll become clear, as in, like, that presentation was a lot about, what are the biological underpinnings of kind of reasons that weights change and things like that. Whereas this is more about, okay, say you have spike time dependent plasticity. What does that mean in terms of what you can encode and how the dynamics of a network changes over time, things like that.
Okay.
So yeah, and then I guess why I care about this. So yeah, as we all have, the driving belief for the Animenta is that there's unique advantages to these biological learning rules, at least in certain cases. And we already kind of leverage, I think, some of these in Monty and of course Numenta's prior work. But then there's scope to expand this. And then I probably won't talk about it today, but because I think it's better after having discussed all the different types of learning rules. But yeah, this kind of unlocks various benefits that that we could realize in Monty. And yeah, I think it's worth kind of saying, so learning currently in Monty is in a sense brain-like in some respects. So I would argue that all the learning we're doing is local. So local, when you talk about like brain kind of learning rules, is basically just that it's information that the presynaptic neuron and the postsynaptic neuron both have. That's all the information you need to determine what the kind of change is going to be. You don't need to transport information from some other distant part of the network in order to kind of update the values in the system. And that's something that Monty already does. And these changes are rapid associative changes. We're kind of strengthening or weakening connections between different parts of the network. They're not necessarily represented as connections, but in terms of like how we kind of associate a certain feature with a location in space, that's kind of a rapid change that's made. And these associations are also sparse in some sense that, like, for example, when we learn an object, we're only storing the locations that we actually care about and the features that are associated with those locations. We're not storing information about any possible location on an object. And I think, yeah, we can agree we want to keep these. So, avoid, for example, introducing backprop in the system, which will start breaking some of these properties. But I think the key thing that's kind of not present, which I do think, could bring significant benefits in the future, and this is something we've discussed before, is high dimensional feature representations and the learning rules that would be associated with those. So the classic example would be SDRs. So this kind of unlocks, using these kind of representations unlocks a bunch of things around kind of more fuzzy matching if you have kind of these representations encoding features or objects. but I'm not going to be talking about kind of what the specific benefits of SDRs are themselves, but rather kind of if you are representing things with a kind of more neural population, what are the kind of biological learning rules that might function in those kind of high dimensional populations? And I won't just be talking about SDRs. In fact, I won't get to SDRs and Numenta's learning rules today. I'm going to start with the kind of more kind of classical firing rates and STDP to give that as a context for Numenta's learning rules next time. Cool. Any questions before I go on?
No, this sounds great. Thanks.
Okay, cool. So yeah, and I'd say the kind of course, when you say learning in the brain, that could be at all kinds of levels. That could be at the behavioral level, and of course, there's biological processes changing the expression of DNA and all this kind of stuff that goes on. But I'm not going to be talking about those really. I'm going to be talking about processes that kind of happen at the level of individual neurons. And I think it's worth I think everyone's probably fairly familiar with this, but just not assuming too much knowledge about like the kind of structure of neurons. So just that it's kind of highlighted in case these terms have come up. So a pyramidal cell is one of the most common excitatory neurons in the brain, in the cortex. It has this kind of distinct structure of these dendritic branches with a long axon that projects and sends spikes out to other cells that receive them. And then the dendritic branches are where the cell can receive inputs, as well as on its soma, which is kind of the body of the cell. And these dendritic branches have the actual kind of synapses on these spines, these tiny protrusions. And so that you can see one here, for example, as like another axon will often kind of pass by. It may produce a small branch, or just directly on it, it may produce the kind of synapse that then connects to the spine. And the kind of knowledge of this kind of structure is key to, for example, Numenta's representation of neurons, where rather than having just like a weighted sum input to represent a neuron, you actually try and represent the structure in the kind of the way neurons learn, where they have apical dendritic segments, basal dendritic segments, and then the kind of feed-forward input at the soma. And so these can essentially, well, yeah, I'll get into this another time, but these essentially can bias the cell. And it's just worth mentioning, if you hear the term kind of segment, that's essentially referring to like a spatial cluster of synapses that are nearby each other in space.
Are you going to talk about the difference between apical and basal?
I will eventually, yeah, not today,
okay.
but
Yeah,
I'll
that's
definitely
pretty
get
different.
into that.
Yeah, because the apical stuff has a pretty different impact, I believe.
Yeah.
And then just a couple of high-level points before we get a bit more into the details. So you could broadly categorize kind of synaptic plasticity or kind of learning changes that happen at the neural level into what are called functional, what you can think of as like weight changes, and structural, which you can kind of think of as wiring changes. And so, if you had these two neurons here that are kind of both projecting their axons to each other, then what you call it, this weight here, if that was increased such that if this blue neuron spikes, the impact on the green neuron is more significant. It's more likely to push the green neuron to then spike. That's kind of represented by the increase in this weight, various biological processes that can cause that. Or you can have more of a wiring change where through, again, various mechanisms, a kind of synapse where one in effect wasn't before, now is. And in general, this is more the kind of flavor of plasticity that Numenta uses. Yeah, partly because, Jeremy talks about a lot of this, which is why I'm not going too much into it, but there's evidence that the fidelity of these weights isn't that high. So like there is definitely evidence of this kind of weight plasticity, but it might be, the equivalent of having like very close to zero weight, very high weight, and maybe two levels in between, if you think of it as kind of a form of quantization.
And I think even in in distal dendritic segments, the impact of the synapse is way lower in individual synapse. So there, it may just be literally binary at that point,
Yeah, right.
whether
Okay.
it impacts at all or not.
Yeah, and then another high level point is just that this is kind of a summary of what Jeremy talks about in case you haven't seen it, but the connection properties and how they evolve over time kind of fall on this large time scale. So if you imagine the kind of connection properties kind of becoming stronger versus weaker, this can happen on the order of kind of milliseconds, the short-term plasticity, all the way up to, yeah, I mean, I haven't tried to map this super accurately, but kind of generally days where you'd have kind of structural plasticity where you're actually growing new synaptic spines. But then in between that, you can modify things about the synapse that give you kind of a kind of medium-term potentiation, so increase in the strength of the weight or decrease, and then a more kind of long-term permanent potentiation. So this would be kind of a weight change, and then this would be a structural plasticity change where you can have kind of a dendritic spine that's sort of a synapse that's inactive, but then through some fairly rapid changes can become active without having to grow a new one. So you can kind of think of these two shorter ones as kind of traces that might serve some purpose in kind of the short term versus more kind of persistent changes that you'd expect to last kind of much longer. And then a final caution, just because I think if you have a background in neuroscience, this is often kind of taken as a given, but you may not have come across this if not. But there's something kind of generally referred to as Dale's law, but basically neurons of a particular type of neurotransmitter, so an excitatory or an inhibitory neurotransmitter, will generally only release that type of transmitter when they kind of connect to other neurons. And so this means that, you know, neurons are either excitatory or inhibitory, and progressive changes to a weight is not, will never kind of flip it from being positive to negative, vice versa.
It's worth pointing out, I don't know if you're going to do it yet, but the inhibitory synapses are quite different. They don't have spines, and they're generally not viewed as participating in learning. I'm sure there's some exceptions to that, but they're sort of more background computational things going on, but they don't.
Yeah, okay.
The rule you were just talking about, spines and forming new synapses, generally are not believed to occur with inhibitory synapses, and they don't have spines.
Yeah. Yeah, okay, interesting. Yeah, I wasn't aware about the kind of spine difference. That's interesting to know.
Yeah,
yeah, another big difference is that it's not, you know, excitatory synapses, I think by and large are very similar, but inhibitory synapses, there's a huge diversity of different types of inhibitory neurons, and they have very different temporal dynamics and very different types of impact on the postsynaptic cell. And so there's like, I don't know, like 5 or 10 completely different types of inhibitory neurons in the neocortex. And
the way I've always viewed it is when you have some sort of algorithm that has to be operated or created in the cortex, those algorithms are often highly dependent on inhibitory neurons, you know, sparsity, synchronization. Those are like the computational background, where the excitatory cells mostly, you know, they are where memories are formed and where you learn things.
Right.
Yeah, that's how I view it too. Like the excitatory synapses are like the memory of the system, and the inhibitory neurons are the control system. It's like actually the CPU, the instructions. It's a very different view than deep learning.
Yeah, I feel like, I mean, maybe at some point that could be interesting to delve a bit more into the different types of inhibitory neurons and as you say, everything from the temporal dynamics and stuff. But maybe there's a recording of.
Yeah, there should be a recording of 1 or 2 you know, videos that we've done on that. It's the data on that is not as clear. There's all sorts of things, but maybe now it's like, you know, 6 years later. there might be more information.
There's a lot of data that's even old about that. I think we said enough perhaps, but just something as simple as, you know, a cell body, the soma of the cell generally does not have any excitatory synapses on it, but it has inhibitory synapses on it. So the excitatory synapses are limited to the dendrites, where the inhibitory synapses are on dendrites on the cell body and also on the axon. So it's a very different type of
Mhm.
effect in the system.
Right. Okay, yeah. So then to get into the kind of broad category, then I'm really going to be talking about Hebbian learning. And I mean, because everyone will have heard of this, but it's this classic rule, associative rule that cells that fire together wire together. And this was proposed by Heb in the 1940s. And I guess one of the kind of clearest demonstrations of this at the cellular level was first done by this Norwegian physiologist in the 1970s, when he kind of showed this long-term potentiation when you cause neurons to kind of spike together. And it's a kind of simple learning rule at face value, but it's worth kind of your kind of immediate concerns that might spring to mind are probably well founded in that, if this neuron is firing a lot and this neuron is firing a lot, and then they keep increasing the weight between them, then where does that end? Weights can grow kind of unbounded in a naive implementation, and it becomes unstable. And so what I think is maybe a useful perspective is that kind of any form of Hebbian learning needs some form of homeostasis to kind of balance the system, and then some form of competition, which kind of connects to what Jeff and Subutai were mentioning earlier about inhibition, in order to be interesting. And so I thought this kind of would be an interesting way to then examine kind of three forms of Hebbian learning, which is kind of the classic firing rate neurons, and then spike time dependent plasticity, and then Numenta's HTM neurons, where all of these, both of these things apply. And so the first two are really kind of more weight plasticity, whereas Numenta's HTM neurons are more structural. So again, I'm sure this is familiar to most people, but just a classic firing rate neuron, just to make sure that we're kind of in agreement on the kind of variables. You have some output, which is dependent on a weighted sum of inputs, where these input neurons are spiking a certain number of times per second. That's their rate. And then typically you'd have some nonlinearity, which ultimately determines the output of the firing this neuron, which could itself be expressed or kind of viewed as a firing rate. And in order for this kind of network to learn with a kind of Hebbian style plasticity, you would update the weight based on the kind of current value plus some change. This is the kind of Hebbian term. So basically the magnitude of the input and the output neuron. And remember, these are never going to be negative. They're always going to be either 0 or positive. And then the learning rate, which is some kind of small factor that slows down that learning. And so, yeah, this is what I was talking about earlier, that if you could just kind of naively implemented this and then had a bunch of input neurons and then an output neuron, and then you wired them up, this weight would likely continue just growing unbounded. And so one kind of popular way that was proposed to kind of deal with this is to kind of introduce this homeostatic term, which was called OHA's learning rule, where there is a negative effect on the weight that is both proportional to the size of the weight and the size of the output neuron squared. And yeah, so the reason I'm kind of going through this is because I think it's interesting to understand kind of what a firing rate can learn to encode before we get on to kind of more complex neurons.
I mean, it's interesting. I'm not familiar with this rule, never heard it before, but the way I always felt it is like, okay, a synapse is just a physical structure. It doesn't get very big. It can only do release so many synaptic vessels at once. So it's like physically limited. But this implies it's tied to the activity of the neuron, which is odd to me. I don't know why that term would be like that.
Yeah, so there's a few ways you can
this equation may reflect what we see in biology, but it's not, I think, what's actually going on in biology. So,
yeah, I mean, you can imagine like a hard bound,
like,
and
it's
that's
limited
also
somehow it's controlled by the rate of firing of the neuron. That's weird.
So yeah, you can imagine a hard bound so that, as you say, like a synapse can only get so big. So you could you can very easily just say, like, okay, weights cannot go above the value of 1.0 or whatever.
Right.
And that's a totally legitimate thing to do, but it doesn't solve all the problems. The other thing is, like, yeah, in terms of what this homeostatic term might represent, I mean, the squared here is, you know, this wasn't a learning rule that was necessarily fit to biological data. That wasn't its kind of provenance, but you could imagine that, you know, every time a postsynaptic neuron fires, maybe it releases some factor that then degrades kind of synapses, like just kind of a decaying term.
I guess from a biology point of view, I've never heard any evidence for that. So it is, again, it may be a useful rule, but I don't think it's reflecting biology as far as I can tell. It's just, you know, synapses are physical structures. They're limited. They have limited area. They can only release so many synaptic vesicles, and they just hit that limit. That's what it is, you know? Okay, but you know,
but I mean, yeah, I'd say most, I mean, even like, I think it's not the same, but the HDM neuron has like a negative,
well, we had a decay.
yeah,
right?
exactly, a decay.
Right. That's not the same as a limiting factor. It's a decay, and there's lots of different ways, you know, but you know,
Yeah, it was, I mean, it was bounded between 0 and 1, I guess, because it's naturally, because it's binary, and then there's a decay that kind of
well,
helps.
it's binary, or it's just limited. So you could say 0 to 50, I don't care. But you know, the synapses come and go, and you know, we do forget, and so there is a part of forgetting is just not, I'm not saying all of it, but a lot of part of forgetting is if synapses aren't used, they just sort of decay, you know, they just kind of go. So you don't want to keep everything active, you know, we forget things a lot.
Right.
Especially my age.
But yeah, so, but yeah, I guess I agree this is probably not the most biologically plausible implementation of this, but I guess it's interesting because OHA was the first person to kind of show that what is the significance of this is that so in this network we just have one output neuron that receives a bunch of input neurons, X, you know, run through XI. And if there's some kind of statistical structure basically to the activity of the input neurons, then the output neuron is going to pick up on that. And in particular, so if you imagine certain presynaptic neurons are firing together, i.e. they're correlated, that's going to, you know, with some random weight initialization, again, all weights here are positive, with some random weight initialization, that's going to cause the output neuron to fire higher when those neurons are firing together. And so those will all experience an increase in their weights up to a limit. On the other hand, if you had some other neuron, not shown here, that you can imagine that is poorly correlated with the others, then this homeostatic term is actually going to kind of negatively dominate. So, you know, the other neurons are driving the output neuron, so Y is high, but XJ in this case is actually low. And so if it started randomly with a non-zero weight, it's going to decrease.
Yeah, there is and this is fine. I don't want to pick apart this, but the basic way, I mean, basically you don't want too many neurons firing and learning at the same time. And
yeah, this is getting before inhibition. I'll get to that in a minute.
Right. Because this is really done by inhibitory neurons, which so it's
well,
not.
But this is kind of imagine for the moment we just have a single output neuron.
All right. Okay, but okay, fine. I can imagine that, but it's not.
Yeah.
We do, right. We do. Yeah. We don't have that. We have these very fast inhibitory basket cells that essentially enforce sparsity. But it's okay. I just don't want anyone to get confused, like, oh, this is what's really going on. It's like, well, it's a good way of expressing some stuff.
Yeah, well, so yeah, so just to recap, so basically presynaptic neurons that are correlated when they're firing, firing together, they're going to experience kind of weight growth to the output neuron, and then uncorrelated neurons are their weights are going to decrease. And the end result of this is basically Y is going to learn to respond to the variation along the first principal component, if that means anything. So if you're familiar with principal component analysis, if you have this kind of cloud of points, some higher dimensional space, there's some kind of lower dimensional structure to that. So this kind of axis of principal of kind of largest variation. And basically that is the dimension on which the kind of Y neuron is going to be most sensitive to, which is kind of an interesting, that's such a
This
kind of
was
simple network.
Yeah, I remember this was a really big deal in the early 90s when we were, when backpropagation had just come out and stuff like that. This was referred to a lot. This was a pretty big deal back then.
Yeah. Yeah. And then, but then what makes it more interesting? So remember, this is kind of the homeostatic term. All you need is that, and then you can kind of get this property. But then to Jeff's point, inhibition is very important. And so anti-Hebbian learning is a term you may hear thrown around. And it basically just means, as the name implies, the opposite of Hebbian learning. So cells that fire together, there's actually going to be a growth in a negative connection between them. So if you now imagine that we have multiple output neurons and multiple input neurons, then any given output neuron, if it's highly active, it's going to inhibit the others. And per Dale's law, it wouldn't, in the brain, it wouldn't do that directly. There would be some inhibitory neuron it connects to first, which is why inhibitory neurons are kind of called interneurons. It would connect to that neuron first, and then when that inhibitory interneuron spikes, it would then inhibit the others. And of course, this is a toy network where you get kind of one winner take all, where one excitatory neuron, when it wins in this output layer, is going to inhibit everything else. And so this is interesting because at the moment, without this inhibition, if you had a bunch of neurons in the output layer, they would all learn the same thing. They would all learn this first principal component. But with this competition, with this inhibition, then basically the first neuron is going to, or one of the neurons is basically going to learn the first principal component. And so it will inhibit the other neurons when it's most active along that component. But then the other neurons can then basically learn to represent the orthogonal principal components. And so in this way, you can get an output. So let's say Y, the output layer, is of lower dimension than the input layer. Then this is, this way you can naturally take like a 100-dimensional input and compress it down into, let's say a 5-dimensional kind of PCA decomposition. Any questions on that? That roughly makes sense.
Yeah, I think, the spatial puller is sort of a variation on this idea, but instead of each neuron being orthogonal to the others, you have an n winner take all, and so it's not, it doesn't have to be, this is one cell wins and they all lose. It's like a subset of the cells win and then another subset wins and so on. So it's not like one cell wins. It's in the brain, we don't see that.
Yeah.
But it's the same basic idea, but it's more of an n winner takes all,
Yeah, and I guess while we're on that topic, like in terms of encoding like more continuous properties, like whether that's like how confident a neuron is that something's in the in its receptive field, or something like that. I guess is that something you guys ever kind of thought about, whether it would be worth having back in something like kind of firing rates or firing probabilities? Or
we didn't have firing rates, but we had the way we would do continuous variables is we would have encoding which was distributed and change slowly with the change in the continuous variable. So you could do that up to some resolution. So we didn't have firing rates as such, but we did have that coarse coding, if you will, in binary. And you do see that in the brain. And we always, I think we always felt the firing rates are just too slow to encode anything useful, because you need to integrate over hundreds of milliseconds to get any sort of, to get a reasonable scalar value from just a single neuron firing. But from a population coding, you can get in 5 milliseconds, you can get a pretty accurate scalar
Yeah.
code.
It's clear in the brain, there are parts of the brain that really are, firing rate dependent. That's a fact. We know that. And there are parts of the cortex that are that way too. But so it's not like this doesn't exist. It's just that when we talk about the kind of representations we form in the cortex, there's much less evidence for that. It's much more evidence for sparse and distributed representations. I mentioned before, like a neuron that drives a muscle fiber to contract is a rate encoded neuron. I mean, the stronger you want to contract, the faster it spikes, and you slow it down and speed it up. The time frames make sense for that. And those neurons are coming, some of those firing rates are coming from the layer 5 cells. So there are firing rate dependent, but when we talk about encoding information in models and so on, the evidence is suggesting it's sparse and it's not firing rate dependent.
Yeah.
Yeah,
There's a bunch of experiments that, like V4 and stuff, you start to see, you see,
within like 100 milliseconds, kind of, you can recognize an animal, and
exactly, or even less than 100 milliseconds. If you think about, the information coming to the retina processing it through a couple of layers, through the thalamus, to B1, and then eventually to B, and there just isn't time for any firing.
I guess the only thing with that is, if you are sampling from like a large population, so like even if each spike, even if each neuron only has time to give one spike, if those neurons kind of are emitting those spikes kind of randomly with +1 statistics or whatever, I think that as long as you sample from a large population, you could still kind of approximate kind of the firing rate input.
Well, you might
sort of that'd still be a population. That's still population coding, but yes, you could still do that. But,
yeah, I see what you're saying. Yeah,
the
but
population.
That means it's not like the same population is encoding completely different scalar values that just by changing their probabilities. There's an actual shift in which ones, and you definitely see that in the brain.
Yeah.
Right. It's so confusing because in the literature, people slide between, someone will be studying a Plasticity of the snail, and they'll be showing these kind of, these neurons that work this way. And the general assumption is like, oh, neurons throughout the cortex of the brain work this way, but it's not true. And so you have to be careful. There's a lot of evidence for this kind of neuron firing, but the evidence in the cortex is quite different.
Yeah, you really need to know which neurons they've been studying. Yeah, and which scenario.
Were you going to say something, Vivian?
I wanted to ask Niels, or really anyone, is there a consensus on which side of the synapse learns? As in, do you learn by gaining more receptors, or do you learn by gaining more vesicles that can emit neurotransmitters, or both?
Honestly, I'm not sure. I think it's a mixture of both, but
well,
I mean, I guess it probably varies too everywhere.
Primarily the postsynaptic cell, but
a big part of it is the area of the synapse itself. How much, like a synapse is just these two membranes that are next to each other at the tip of the spine, and there are vesicles containing the neurotransmitters, and they pass across that gap. And how many can pass across that gap at any moment in time is dependent on a bunch of things, but certainly the area is important. So a big, well-learned synapse will have more area. These are really tiny areas, of course, but more area than a skinny little synapse. So that's one thing. Another thing is it's a very complicated area, because when the vesicles go across and they release their neurotransmitter, they have to be replenished on the presynaptic side. And so, there's not an infinite supply of them. So it's, there's a synapse that fires, meaning it passes its neurotransmitter. It has a period of time where it can't do any more, or can't do much, because it hasn't regenerated new vesicles on the other side. So how quickly it can replenish vesicles. So it's a complex equation, but mostly it is viewed as learning is on the postsynaptic side. That's the general rule. Synapses get bigger, mostly on the postsynaptic side. You can see the spine's getting bigger. And that's, I would say most of the learning was generally viewed to be on that side, the postsynaptic side.
And I was just asking to sort of like, because with Rojas's rule, so did I get the rule name right? It assumes information's flowing back to the synapse, and that's going to be easier if it's postsynaptic or presynaptic. But
Right. So all of these rules, at least our rules, require in the HTM neuron, it requires that there is a back action potential, meaning once the cell fires, the spike goes down the axon, but there's another spike that goes back up the dendrite, and it reaches the point where the dendrite was active and was receiving information, and that is the learning signal. It says, hey, yeah, I did fire. Therefore, this was useful. You guys should strengthen this. If a
you've got a
synapse,
way to do coincidence detection at the synapse.
right? Otherwise, you wouldn't know. These synapses are not at the soma. Remember, they're not at the body. So, and then most of them are pretty far from the cell body. So they need to know if the cell spiked. And so there has to be this back action potential.
Cool. Yeah, so I guess just kind of the final note on this is just that, yeah, I brought up OHA's learning rule because it's kind of a classic example of how to learn in these networks, but it's not the only way. So yeah, as mentioned, you could kind of have a hard bound on weight values, which, there's plenty of biological reasons that might exist. And this would be kind of clipping if you want to use kind of more machine learning terminology. And then I think, one other classic learning rule that's worth just mentioning is this BCM learning rule, because so the OHA's learning rule, I mentioned it learns the principal components, but actually if you look at the kind of receptive field properties of neurons in V1, say, they don't correspond to the principal components of natural images. Instead, they correspond to local feature detectors like oriented bars, or at least that's what it looks like when you kind of examine them. And this BCM learning rule actually learns more like these, and it's not that different from OHA's learning rule. It's just a bit more complex in terms of how the weight changes are dependent on the firing properties of the postsynaptic neuron. So yeah, then with that, I was then going to talk about spike time independent plasticity, because this is where you start to get into how kind of weight changes are actually happening in the brain. So before, this is kind of a high level approximation of what we kind of seem to observe. This is much closer to kind of mathematically how synapses actually change in the brain over time. And it gets its name because of this kind of relationship between, so if you have a presynaptic neuron arriving at sending a spike arriving at a postsynaptic neuron, so this is the presynaptic spike and the postsynaptic spike. Basically, if this one precedes that one, then you will get this long-term potentiation, the increase in the efficacy of that synaptic weight. And if the ordering is reversed, you'll get a decrease. But this depression in the efficacy of that synapse. But this only happens within a narrow window around the kind of timing of the two spikes. And so essentially, yeah, the output neuron is then looking not just at whether I'm firing and you're firing, but really it's more of a causal kind of learning rule, because the output neuron is kind of saying, is there evidence the presynaptic neuron was actually responsible for my spiking? Which personally I think is a pretty cool thing that that evolution kind of figured this out, and makes a lot of sense when you think about the amount of noise and kind of random spikes that are happening in the brain, that you would want this kind of sensitivity. And
this is another way. This is another way that neuron activity gets separated. We talked about inhibition. Basically, you're going to say, hey, if your input couldn't have possibly been responsible for my output, then I'm going to tell you forget that. And so it will separate out neuron representations through this rule as well.
Yeah. Yeah. And I guess, yeah, I mentioned that, this is kind of actually how weights are being changed. So I just thought it was worth kind of showing some actual data from biological experiments where they're showing the kind of percentage change in the efficacy of the synapse as a function of kind of when the pre and postsynaptic neurons spiked and the kind of difference between those. And you see it forms this kind of curve that is so often shown quite nicely. And so this is kind of essentially then a coincidence detection learning rule, kind of a temporal learning rule. And it means that you can have output neurons that are sensitive to the timing of input neurons, and in particular, kind of neurons that are all going to have signals that arrive together within a short period of time. And so this is a computational study from kind of one of the groups that's been most interested in this kind of computation in the brain. And so what they're showing here is, so along this y-axis, you have the neuron IDs. So neuron 100, neuron number 1. And these are all kind of in the input layer. And then the blue dots are when they spike over time. And their kind of spike times are approximately random, except for these patterns shown in red, where they will fire with some kind of fixed, kind of within some short time window relative to one another. And so what's kind of important to note is that if you look at the firing rate of the population, that's what's being shown here in this bottom box. So if you kind of sum along this axis, the population's firing rate never changes significantly, even when these patterns are being presented. And similarly, if you look at the firing rate of each input neuron across the simulation, these aren't any different from the neurons that aren't part of the input pattern. And so basically, a neuron that is limited to a kind of simple firing rate code and firing rate based changes to its weights is not going to become sensitive to this input pattern. But a neuron with spike time dependent plasticity does. And so it just basically means that you
Can you repeat again how they had identified the red neurons? Because sometimes it's just one of them, so it doesn't look like there was actually a rate that changed, like neuron 50, for example, there's just one red dot. So I don't see like why there's a change in the firing pattern.
Neuron 50.
Yeah,
You mean like here, sorry?
Yeah.
Wow.
So the firing rate is taken across the entire period.
Yeah.
Like 600 milliseconds. So that's what's measured here. So like, okay, it's like this one is fired 1, 2, 3, 4, 5, 6, 7, I don't know, 8, 9 times or something. And then this firing rate is calculated by looking across the different neurons, how many are spiking at that point in time.
Yeah, but what decides if it's red or blue?
So they're inserting these patterns. They're coloring it red to show us that pattern is there. But it's basically they're just saying these neurons are part. Yeah.
So did they artificially increase the synchrony during those?
Yeah, they basically artificially inserted this pattern so that these neurons are kind of firing with some distinct pattern. Like if you look, there's kind of even these like subtle details, like these 3 neurons firing with this offset and stuff like that. But the statistics of their firing, like if you just look at the firing rate, is the same. It's not being changed during the presentation of these patterns. So it's synthetic data, basically questioning like, okay, if you look at the population and it looks like random noise, but secretly there's actually these temporal patterns happening, would a neuron with STDP learn to respond to those patterns?
Okay, so they know that at the red points they artificially changed it.
They know that, but the neuron doesn't know that.
Yeah.
It's an unsupervised learning.
Yeah. Okay.
Cool. But yeah, no, good question. Yeah, so I should say, yeah, everything I'm talking about is unsupervised learning, as well as, yeah, local. Um, so, yeah, anyways, but so an STD, neuron, an output neuron, a spiking neuron with STDP learning rules does learn to respond to these patterns. So that kind of, what that suggests is that if you equip neurons with these, this kind of more biologically plausible learning rule, it opens up the kind of capacity of what kind of information, how information can be encoded, where you're no longer limited to just firing rates, but you can respond to kind of particular patterns in time as well. But
Are
I
they
think,
saying that if the neuron learns to respond to this within the red thing, if you were to just scramble the red patterns, it would not respond anymore?
so I don't know if they made that claim, and I
Because
think
I
the
don't think
amount of
that would work. So it's not really sensitive to the timing of the red.
yeah, I think you could do both. Like I think it would be fairly straightforward to set the like time constant so that you would be insensitive to their precise order, so that you could have like quite a lot of jitter, and it's more that they're kind of synchronous within like a, let's say, 20 millisecond window. Or you can have time constants so that they have to fire, within 3 milliseconds of this prescribed pattern, each neuron.
I guess I'm just like, suppose you did exactly what they did here,
Yeah,
right? And the claim is, okay, it's sensitive to the timing of the input neurons.
yeah.
But after this learning process, if you were to scramble a few of these the timing of a few of these red dots, I think it would still fire. It's not sensitive.
I think that's what I think it does depend on the time constant. So like,
Okay.
yeah, like, especially,
But
and
each
I was going to maybe
synapse
talk
has
about
its
this
own
time.
time constant? Does each synapse have its own
No, but
time
as in
constant?
like, so if the time constant's fairly wide, then I agree. Like, and sorry, I mean, yeah, I guess there's the STDP time constant, which is already fairly broad, but more I'm thinking the like conductance and things like that, where basically like the time over which the spikes can be integrated by the postsynaptic spiking neuron. And
Yeah, but I don't know if that's true here. Because if you look at just those, you pointed out that diagonal pattern here. If
yeah,
instead it went like this, I don't think it would make any difference to the output, regardless of,
yeah, not
I mean,
in
in
this
this exact
experiment, but I
scenario.
think there
Yeah.
is a setting where that would. So I
Oh
was
yeah,
going to
yeah.
maybe talk about this then the next time with
Right.
like,
You can construct that, but I don't think STDP enforces that. I think the claim here is that STDP causes neurons to become sensitive to the timing, but that's not true,
no,
I
yeah,
don't think.
I would say that that's not true. Yeah, yeah. Basically,
Yeah.
you'd need like heterogeneous conduction delays,
Yeah. So you can do it, and
and
then you don't
basically
need
like
STDP
if
anymore.
well, you still need STDP, but yeah, STDP is no longer like sufficient.
Yeah, yeah,
Um,
exactly. Yeah.
yeah, so like, yeah, just for anyone else who's curious, I guess, and yeah, I'll talk about this next time. But this is kind of a form of synchrony, although yeah, they have kind of fixed the pattern a lot. But there's another kind of called polychrony, where if let's say, yeah, they're firing with this kind of specific pattern, if you have asynchronous or like heterogeneous conduction delays all landing on your postsynaptic neuron, depending on the values of those delays, these might all be arriving within a very narrow window, like, within a couple of milliseconds of one another. And then you can imagine potentially an output neuron that's then sensitive only to this pattern. And then if you reverse the order of these and all these and whatever, like they all fire within this kind of time window, but in a different order, then that output neuron would not respond. But that's a big if, because you need those
Yeah.
delays to be correct. But yeah, I think it's worth kind of tying it back to this kind of general principles of Hebbian learning. So although now things are kind of dependent on time, it's still fundamentally a Hebbian kind of learning rule. So you still need some kind of form of homeostasis. So again, you can have this issue where weights will grow kind of unbounded. And so one of the first papers to kind of computationally study STDP, they basically had this kind of factor where inhibition slightly, oh, sorry, I should say, so this is kind of how much the weight is going to change depending on the difference in the timing of the spikes. And basically, you either have like the weight being increased, in which case there's this kind of positive learning rule, or learning rate, or it's the weights are being decreased. And then this just is determining kind of the sign and the magnitude of that for these curves. But basically they made the inhibitory kind of learning factor or the decrement learning factor larger, so that it kind of balanced learning in the network and it didn't kind of run away. Again, it's not necessarily good biological evidence for that, but it's just an example of the kind of challenges and things to be aware of when kind of, I guess, working with Hebbian networks. And actually, for like a real Hebbian network that's still, I mean, these are very, real spiking neural network. They're generally very challenging to get to learn anything. And so, in this study, it was still relatively simple stimuli. I'll show in a couple slides, but they had a variety of kind of biologically motivated, so they were all local and everything like that. But it was like 4 different homeostatic factors that were all working in concert to get stable learning, and so that they could basically have the learned representations maintain over a kind of unbounded amount of time, even though there was kind of constant noise being fed into the network. And similarly, the competition is still important. So this was a follow-up study from the muscular group to the early one I showed. And here with this first one, they were basically showing that a single spike neuron could learn this pattern. It could learn a single pattern. So kind of the equivalent of the single firing rate neuron learning a single principal component. In this follow-up study, they showed that multiple output spiking neurons could learn multiple different input patterns and become sensitive to those. LIF just stands for leaky integrated fire, which is a type of a simple mathematical model for a spiking neuron. That's that these output neurons learn different patterns. So yeah, I mean, in general, this seems pretty neat, and there was a lot of excitement about kind of temporal code in the brain. I'd say particularly between 1996 and 2006. Tons of papers about this in kind of the biggest journals there are. But maybe around 2007 onwards, there was this kind of general shift in the zeitgeist where there was concerns about kind of plausibility in terms of kind of noise in the brain and things like that. And I also wonder to what degree maybe the success of artificial neural networks as models, or the embracement of artificial neural networks as models of the brain, kind of dampened enthusiasm around kind of a temporal code kind of being significant. But I think you can definitely say is the temporal sensitivity of STDP is a biological fact. And so the existence of temporal codes in the brain perhaps less clear, but I think kind of, when Jeff alluded to this earlier, there's, it's probably somewhere in between. So what's interesting actually about STDP is that they themselves are kind of a hybrid. So this is the study I mentioned earlier that had like 4 different homeostatic factors. These were the kind of stimuli. So if you imagine this is a 64 by 64 grid of neurons, the white neurons are firing with a higher rate, and they will be showing these different inputs. But the spike times of those neurons are governed by a Poisson distribution, so they are kind of random in time. There's no kind of synchrony or anything like that going on. And this neuron that's receiving these inputs is a spiking neural network with STDP-based learning. And it will learn to encode these different patterns. And in general, this connects to the fact that essentially at low firing rates, STDP is in general more temporally sensitive, whereas at high firing rates, it starts to act more like standard firing rate Hebbian learning. And part of the reason this emerges, I just thought of this to mention this, but I could have had a better graph. but you can imagine you have this neuron spiking and then this neuron spiking. But now imagine that this presynaptic neuron is firing so frequently that it fires at this time as well. So just after the other one. So it's firing just before and just after. And that's generally the kind of regime that starts happening when you have really high firing rates, where it's kind of symmetric. And we know in biology that in that kind of regime, like if we experimentally measure the neurons, they tend to act more just like the simple Hebbian weights. But then, yeah, as mentioned, at lower firing rates, these two are going to be very different, whether the spike happens before or after. And so then you are going to see this more asymmetric, temporally sensitive changes to the weights.
You generally don't, I mean, there are neurons that spike very rapidly, but that's not typically, for example, pyramidal cells. They, there tend to be more inhibitory cells. So it's not clear that particular thing we're talking about actually occurs very often. Most neurons don't fire that fast. That role, this idea would come to play. But some inhibitory neurons do. In some places you see that, but it's pretty rare in the cortex. You do see a bunch of neurons that they don't emit a single spike, but when they become active, they emit a train of spikes, anywhere between 3 and 5 long, and those are very close together. It just complicates things.
Right. Yeah, I was going to maybe try and talk about bursting next time,
Right,
because
right.
I know that's something you guys account for.
Right,
Yeah.
because that can lead to these what they call metabolic changes, which are changes in the chemistry or slower changes in the cell. Anyway, yeah, I don't know if I should keep interjecting my biases into this. It's a good presentation.
No. Yeah.
Yeah.
I mean, I've always found that STDP was over, you said had its decade and then it kind of declined. There's a very, very simple explanation, which you've already given, is that there's a lot of time delays and uncertainty in the brain, and a lot of neurons have a background firing rate, which is pretty low, but they're kind of firing a lot. I mean, they're firing all the time at some low rate. That's pretty common. And so there's a lot of random spikes that don't mean anything. And so this rule basically says, you know what, let's only pay attention to the ones that are truly causal, that this predicted this, so if presynaptic neuron is a good predictor of the postsynaptic neuron, well, we want to remember that. And it shouldn't be much of a delay because we're not trying to predict, we're not trying to look back in time. We're just like, right now, these neurons, does this one happen before this one, and it just sort of cleans up the whole learning rule stuff. And clearly if something happened after, if the presynaptic neuron fires after the postsynaptic neuron, it couldn't possibly have predicted it. So forget that one.
Yeah.
There's very simple pedantic mechanics here that could say this is why the brain works like this, as opposed to this is an important part of how information is encoded. It's more like, hey, it's an important part of how synapses learn, but I've always viewed it as sort of not really an information theoretic component.
Yeah. Yeah, right.
But as you point out, a lot of people wrote a lot of papers suggesting otherwise.
Yeah.
So.
Yeah, so yeah, I guess it's also just kind of on this note of like, okay, maybe it's not like a black and white in the brain. I think it's also worth mentioning that there's some neurons that are at least more amenable to encoding information in more kind of temporal-like activity. So, for example, like the spikes that arrive from thalamic neurons, like LGN, onto the cortex, tend to be more kind of driving inputs that have where just a handful of spikes can be enough to cause a postsynaptic spike, whereas many other spikes in the brain you might need hundreds to kind of come together. And in general, like you can kind of show that these kinds of small like sparser high magnitude spikes, if there was kind of a temporal code, it would you would be using those kinds of synapses to kind of encode it and transfer that information.
we proposed in our neuron paper an alternate explanation for that, which is that the vast majority of synapses are pretty far from the soma, and they're on the dendrite branches, and any individual spike out there has very, very little effect on the soma. So what typically a researcher will do, they'll measure the voltage inside the body of the cell, the soma, and they'll see what happens when a presynaptic neuron fires, and they'll say, hey, this presynaptic neuron fired, but it had almost a very tiny little effect on the soma. Therefore, we're going to need hundreds of them. And other ones will have a very large effect on the soma, so we only need a few. But we propose an alternate explanation, which is the ones that are far from the soma, you don't need hundreds to summate. You just need 15 or 20 near each other. And then they generate this dendritic spike, and then that dendritic spike has a large effect on the soma. And so the vast majority of the studies that people did in this area, they didn't account for that possibility. They just measured like, oh, well, this synapse doesn't seem to have much of effect, therefore we need a lot of them, as opposed to, well, what if we had 15 of them at the same time in the same segment of the dendrite, then you'd have a very different effect. So I wanted to point that out, that that was a big part of what our neuron theory says. It explains that difference. It gives a functional reason for it.
Right. So yeah, just that I guess we shouldn't discount at least the possibility that binary, temporal, and kind of more sort of binary codes could be working together. But
right, I think before we get on to this, one of the temporal codes that we talked about recently is this idea of groups of neurons firing in sequence during a phase of a background frequency, you're looking at the theta frequency within the 100 milliseconds of or whatever it is of that cycle. Neurons go through a sequence of patterns, and that's a type of temporal code, which is obviously very important in the brain. But it's not the kind of temporal code that most people think about. It's, most people think about, I believe what you're talking about, most people think about temporal code as like the exact timing of individual synapses, or individual cells, where clearly that's important in some parts, but it's much less evidence for that in the cortex.
Yeah.
Maybe the way to put it is that everything you said here is correct, but it's also very, very confusing.
Yeah, hopefully not too confusing.
Well, you presented it clearly, but I think, a lot of people, a lot of the research out there hasn't really incorporated the kind of the insights that we've had. And I don't sit there and say, hey, why aren't you guys listening to us? I don't feel that way at all, but I do feel like we really understand some of this pretty well. And the vast majority of researchers out there don't think about it that way. They just don't even consider these possibilities.
There's a lot of really detailed experiments on plasticity and that have very specific types of plasticity rules that I found correlate really well with what we did in the neuron paper. I have this detailed list of things, just like that P- that you pointed out, for example. In that paper, there are experiments that validate that. And then what happens when a dendritic segment fires and the postsynaptic cell does end up firing, or it doesn't? What's the impact on the dendritic branch? There's specific things that happen there that correlate really well with our learning rules. And then there's some there that we haven't yet incorporated, I think. But there's a lot of really detailed things that are quite important at the end of the day. I feel like this level is interesting, but it's hard to know what to really make of it until you actually put it in an algorithm, actually put it in a network and have it do something real. And that's the piece I feel like, I don't know now, but at least a few years ago in the computational neuroscience community, they didn't do much of that at all. They didn't show how you could combine all of these to recognize an image of a cat, for example. Right. Or, they show, okay, oriented bars emerge, okay.
Yeah, and I guess on that note, so like, yeah, just kind of summarizing. So, yeah, these kinds of two Hebbian learning rules I talked about today. So the kind of firing rate based ones, they can learn input features like the PCA of the data, or more local features, which I didn't get into, like kind of oriented bars. And then STDP based kind of spiking neurons are essentially like a superset of this. They can learn temporal spike patterns, but they can also learn what kind of firing rates neurons can.
Yeah.
But
Yeah, go ahead, sorry.
oh yeah, but to your point, Subutai, they're extremely complex to set up and maintain and just get to be stable. Like yeah, I think in general, it's a great way to start pulling your hair out, is trying to work
Yeah.
with spiking neural networks.
If I believe, I think we shouldn't be tying Hebbian with firing rates. I believe Hebb did not talk about firing rates. He just talked about, firing together. And
Yeah, sorry, I didn't mean to imply that it's
right.
Yeah,
only for
it's-
that.
We use Hebbian learning in our neuron rule. I mean, but
Right.
we're not using firing rates. I just want to be careful that those aren't really
Yeah.
tied to-
Yeah, it's really interesting to read what he actually said. He did not say neurons that fire together, wire together, as an example. He had a
Right,
very
right.
specific sentence or two that's very telling. And it's
Right. Yeah, I can't remember
it's
what it is,
interesting
too.
to read that, because it's it's a lot more precise, I think.
And if you don't mind, I'd like to make a comment back to the question Michael asked about earlier.
Yeah.
Think about a synapse, right? You have an axon and a dendrite, and they might be near each other, and they may not have a synapse at all, but they're near each other, and it's sometimes referred to as a potential synapse. And so, even in that situation where the axon and the dendrite are near each other but not connected in any way, they will, a Hebbian learning type of rule, they will form a synapse. So they will grow a new synapse. The brain has the ability to do this. It's, I know you're not touching, but you should be, and there's these other neurons or these other cells in the brain that facilitate this. And then you start off with a very skinny little synaptic spine. It's really like like a little thread, and it works. And as the synapse trains, that it gets reinforced, that spine gets bigger. Now, we talked earlier about when it gets bigger, maybe more neurotransmitters get passed through. But there's a lot of evidence, and some other people wrote about this, and we adopted it, that once the neuron gets to a certain size, it doesn't really, and you still train it, it doesn't really pass any more neurotransmitters. That is, its weight value doesn't go up, but the spine gets bigger. So the spine continues to grow, even though it's not transmitting any more synapses. And that the idea behind that is that the spine is a sort of permanence. It's like how hard is it to forget this synapse? Once you've built it and it's structurally there, it's a lot harder to get rid of than if it's just some skinny little thing. And so we adopted that as we called it permanence. It's a learning that is not about increasing efficiency or transmission, but more about permanence of memory. And so things that have been reinforced a lot, even if you don't use them in a long, long time, you'll still remember them. Where something that you learned recently, and it's just as it has just as much effect as the old synapse, but you might forget it pretty quickly if you don't continue to reinforce it. So I just wanted to point out that's back to what Michael asked earlier. Learning is not just about increasing efficiency; it's also permanence of these memories.
Yeah. And so the presentation from Jeremy, there's a lot of discussion around the kind of biology of that, and then otherwise the next presentation I give, I'll go into, I guess, Numentz's actually algorithm for how the permanence values change and things like that, and then what the significance of that is.
Could you just, I mean, you had the link in the presentation to that.
Yeah, I can share it on
just
Slack.
put it on Slack someplace. I'd like to listen to it.
Right, all the way. Start. Yeah. Yeah, it was 2 presentations from November 2021.
Oh, yeah.
But yeah,
okay.
I'll send those in Slack.
Jeremy Forrest, I guess I could just look at.
But yeah, I just wanted to kind of, in summarizing, just say that, yeah, just kind of the kind of one thing to just remember is that, yeah, at its core, like Hebbian plasticity is kind of working in both these firing rate and these spiking networks. And so some homeostasis and some competition is needed. And actually, when we talk about Numenta's algorithms, the same will still apply because it's still, as you were saying, Jeff, fundamentally Hebbian plasticity as well. So yeah, so that's what I'm gonna focus on next time, is this kind of structural plasticity and the role of the I shouldn't say and the role of dendrites. But there's a few other things I'd like to just touch on briefly. I mentioned kind of myelin or polycrity and how that might connect to myelin plasticity. I think kind of dopamine and serotonin is worth kind of referring to briefly. And then, yeah, some other things we've talked about recently, like contrastive learning, representational drift, predictive coding, and then maybe local-ish backprop in the brain. I think it'd just be interesting to touch on. But hopefully this was like a useful starting point.
Yeah.
Yeah, thanks. That was great.
Cool, anyways.
Yeah, thanks, Niels.
Yeah, then I've got these slides about kind of potential benefits, but I think they'll make more sense after we talk about the other learning rules, and those are all clear.